we'd like to start with just defining what pathology is. Pathology is basically the study of disease. And in the study of disease, we consider four different things. Basically, four aspects of the disease process. We discuss the cause of disease, which the term we use for that is etiology, which means cause. And we can look at different kinds of causes, like intrinsic causes. These are hereditary or genetic causes of disease, versus acquired causes, things that we acquire later on in our lives. We also talk about the mechanism of disease development, which we call pathogenesis. That is, how does the etiology or the cause of the disease lead to the manifestations of the disease which we can see and feel? That's called pathogenesis. The next thing we consider is the structural, the physical changes that happen to our cells and our bodies in a disease process, and we call those morphologic changes or morphology. And we can observe the morphology both grossly and under the microscope in order to get an idea of what disease process is going on in a particular individual. The last is the clinical changes or the functional consequences of a disease process. What does somebody with this disease look like? What happens to them? What clinical manifestations, what symptoms and signs do they have? Now, it's important to distinguish between symptoms and signs. Symptoms are things that the patient feels. I feel tired, I feel sick, I feel whatever. Those are symptoms. Signs are things which the examining physician observes. Tachycardia, hypertension, and so on and so forth. Fever, whatever. Those are signs, things that we observe and measure. And so those are symptoms and signs, and those are some of the functional consequences of a disease process. Pathologic processes generally occur as some response to stress. And so in order to understand pathology, we have to understand how the body responds to stress, how it does normal adaptations, and how those adaptations can go awry and cause disease. Now, our organs are made up of cells and extracellular matrix, and virtually all the forms of organ injury begin with some structural or molecular alteration to those cells. Now here's a schematic view of what that might look like. So we have a normal cell, and the normal cell goes through normal homeostasis, is maintaining itself at a normal level. Now there's two things that can happen. One is there can be some kind of stress, and a stress is anything that requires increased demand on that particular cell. Now, cells up to a point can adapt to that stress and form a new steady state, and that new set point for homeostasis. But at some point, the stress may become so extreme that the cell can no longer adapt. And when that happens, the cell becomes injured and eventually dies. Now that's one pathway to cell injury and cell death. The other pathway to cell injury and cell death is through direct injurious stimulus to the cell, direct damage to the cell. Whether it's cut or burned or whatever, that direct stimulus bypasses the normal adaptation mechanism to cause cell injury and cell death. So let's talk about different forms of adaptation to stress. One of the ways that our cells adapt to stress is by increasing the number of cells, and we call that hyperplasia. And that's caused by cellular proliferation. Cells divide and become more numerous. And this often involves stem cells. These are the cells which are still cycling in the cell cycle and responsible for renewing cell populations. Now, hyperplasia can be physiologic or pathologic. For example, the uterus during pregnancy, there's a hyperplasia of the endometrium. And that's a normal physiologic response required to maintain nutrition for the growing fetus. So that's a physiologic or a normal hyperplasia. Now, it can also be pathologic. In certain conditions, the endometrium may increase in size. We may get an endometrial hyperplasia in response to some stimulus, and that can be pathologic then. Can lead to excess bleeding or can eventually lead to cancer. Now remember, pathologic hyperplasia is a soil for cancerous proliferation. Anytime you have hyperplasia beyond normal physiologic mechanisms, you always run the risk of developing neoplasia, which we'll talk about in a different section. Now another adaptive mechanism is what we call hypertrophy. In hypertrophy, we don't increase the number of cells, we simply increase the size of cells. An example of this is muscle hypertrophy. Somebody who has hypertension, for example, and has to pump against a greater afterload, their heart will adapt to that by increasing the size of the muscle fibers so that they can squeeze harder, so that the heart function returns to normal. In this case, the number of muscle cells doesn't increase because it's a dead-end population. Those cells can't divide anymore. So the only way to adapt is to increase the size, increase the number of protein, myosin and actin fibers in that muscle tissue. So that's hypertrophy. So hyperplasia and hypertrophy can usually occur together. But if the adaptation is occurring in a cell, for example, a cardiac myocyte that can no longer divide, then you get hypertrophy alone. Here's an example of what that looks like. So here's a normal cardiac myocyte, normal muscle, and you put some stress on that muscle. Okay? So let's say, for example, that hypertension puts some stress on that muscle, and the muscle needs to increase its functional capacity in order to meet that new demand, then it hypertrophies. And it can do that to a certain point. That's a normal adaptation to a stress. Now, let's say that there's a cell injury instead of just a simple cellular stress. Let's say a hypoxic injury that we get in myocardial infarction. In that case, you get a direct injury to the cell, which leads to, rather than hypertrophy, leads to cell death. And so those are the two pathways that we discussed before. Now, another response to stress is what we call atrophy. And this is a decrease in the size of cells due to disuse or to loss of input or to aging and senescence of those cells. And what you see in this picture is an example of a normal brain on the right, or labeled B, and a brain of an individual with dementia, with Alzheimer's, in A on the left. And in that case, there is a loss of cellular tissue that leads to atrophy. Now, you see the same thing, for example, in skeletal muscle. If you don't use a muscle for a time, it shrinks in size because the demand which led to that adaptation has gone away, and so you get atrophy of those cells. Now, the mechanism of atrophy is as follows. There's the key thing is the degradation of protein. And so excess protein that was built up in a hypertrophy, for example, when you get disuse or loss of input, all of that protein gets broken down, and that's what leads to atrophy. And the ubiquitin proteasome pathway is important in this. In that pathway, you get labeling of proteins by a small molecule called ubiquitin, and that signals those proteins to be broken down by a protein complex called the proteasome. What you typically, another thing that you can see in atrophy is autophagic vacuoles. These are vacuoles in the cell that are consuming, they're like secondary lysosomes consuming cellular proteins so that the cell will atrophy. Now, another adaptation to stress that's pathologic is what we call metaplasia. Metaplasia is a reversible change in which one adult cell type is replaced by another cell type. An example of this is what we get in Barrett's esophagus, for example. Somebody that has long-standing gastroesophageal reflux disease can get so much injury to the esophageal lining that you get a change from the normal squamous epithelium to more of a columnar epithelium. You can also see that in a disease we call myositis ossificans, where you get formation of bone within muscle. So you get conversion of one terminally differentiated tissue into another type of terminally differentiated tissue. Now, this comes about because of some reprogramming in the stem cells, which are responsible for generating that tissue. Now, metaplasia is a pathologic process. This is not a normal physiologic mechanism, but a pathologic one. And in fact, metaplasia is in many cases a precursor to cancer. Here's an example of metaplasia. This is Barrett's esophagus. You see on the left normal squamous, stratified squamous epithelium. And you can see on the right-hand side, the stratified squamous epithelium has been converted to glandular, columnar, more intestinal type epithelium. That's metaplasia. Now, dysplasia is one step further. Dysplasia is conversion of a normal structural tissue to one that is deformed. Cells that have lost their uniformity, lost their architectural orientation. For example, we see dysplasia of cervical epithelium in human papillomavirus infection. The normal squamous epithelium of the cervix gets disoriented, becomes pleomorphic, loses its cellular uniformity, and starts to look very unlike normal stratified squamous epithelium. Now this is caused by a persistence of that same stress that caused the metaplasia in the first place. So whatever stress it is that caused that cellular change persists and leads to dysplasia. And in the example of cervical dysplasia, it's a prolonged human papillomavirus infection, for example. Now dysplasia is the last step before we start getting irreversible or malignant transformations. In other words, cancer. All right, so those are all adaptations to stress. Some physiologic, like hypertrophy or hyperplasia. Some pathologic, like metaplasia and dysplasia. So that's one pathway. Now what about direct injury to cells? And you see that on the right-hand side of this schematic, where you get an injurious stimulus, a direct injury to the cells. Now there are a bunch of different causes of direct cell injury. One of the common ones is hypoxia. Remember that we have to have oxygen to maintain the metabolic processes in our cells. So if something happens where our oxygen is lost, then this causes cell injury because we lose our aerobic oxidative respiration. We lose energy that are required to maintain normal cell processes. Now, hypoxia can be caused by ischemia. That's when we have blockage of a blood vessel so that we have loss of blood supply. It can be caused by cardiorespiratory failure, in which we're able to oxygenate blood, and the blood we are able to oxygenate, we have difficulty circulating, and so that decreases oxygen delivery to the peripheral tissues. We can also lose the oxygen carrying capacity of the blood. For example, somebody that has anemia, that has a decreased number of red blood cells, has decreased oxygen carrying capacity. So these are all things that cause hypoxia. Another thing that can cause cellular injury is physical agents. Direct trauma, for example. Thermal injury like burns. Radiation damage, electric shock, so on and so forth. Anything that causes physical damage to a cell. Here's some other things that may cause cell injury. Chemical agents, for example, that disrupt the chemistry or directly injure cells. Drugs can do that as well. Infectious agents, we talked about the response to HPV, for example. These are things that can cause cellular injury. Immunologic reactions can cause cell injury. There are congenital and genetic derangements that can cause cell injury. And nutritional deficiencies or nutritional excesses can also cause direct cell injury. Now, the mechanism of cell injury depends upon the duration of the exposure, the severity of the exposure, and the ability of the cell to adapt. For example, in hypoxia, neurons are very sensitive to hypoxia. A neuron that goes without oxygen for 3 to 5 minutes may reach irreversible damage and die. Whereas for a myocardial cell that has more of a reserve, that may be 1 to 2 hours. And it may be even longer for skeletal muscle. So depending upon the adaptability of that cell, it may be more or less likely to be injured. There are 4 systems that are especially vulnerable to this type of cell injury: membranes, aerobic respiration, protein synthesis, and preservation of the genetic apparatus. Those are the 4 systems that we are concerned about in physical injury. And in fact, damage to one of those systems may affect the others and cause a cumulative or a chain reaction. Let's talk about a couple of examples of injury. Let's start with hypoxia. What happens in hypoxia? Well, the first thing that happens in hypoxia is that it shuts down the electron transport chain. Remember, if we can't run the electron transport chain because we don't have the oxygen to be a final electron acceptor in that system, this diagrammed here, we lose our proton gradient across the inner mitochondrial membrane, and we can no longer make ATP by the ATP synthase protein complex. And so we start to lose ATP right away. Now, loss of ATP is going to have 2 different consequences. One is that rather than generating energy from aerobic processes, now we convert completely to anaerobic metabolism. Remember, when we do anaerobic metabolism, we're just doing glycolysis. And so at the end of that, we end up with a whole bunch of NADH and a whole bunch of pyruvate that we have to get rid of. And that's done through the formation of lactate, but that leads to lactic acidosis. And that decreases the cellular pH, which further damages the cells, further breaks down the metabolic processes of the cell. The other thing that happens when we lose ATP is we lose the osmotic gradients that are required for the proper structure of that cell. And that's because the sodium-potassium ATPase, an energy ATP-requiring protein, is no longer able to function. And so the normal ion gradients that we maintain between intra and extracellular compartments is lost. And this may cause cellular swelling or shrinkage and consequent cellular dysfunction or death. Now, if oxygenation is restored within a reasonable amount of time, these changes are reversible. And here's just a schematic of what we've talked about already. Decreased oxidative phosphorylation. So you get ischemia, which leads to decreased oxidative phosphorylation, which decreases the amount of ATP, and that causes the ion gradient problems, the anaerobic glycolysis problems, and some other effects as well, including detachment of ribosomes, decreased protein synthesis, and lipid deposition that all occur because of a loss of ATP. So here's what the reversible damage of hypoxia looks like. This is EM figures of renal tubular epithelium. You'll see on the left that there's nice, small, dark mitochondria. There's a nice villous border to the cell. But what happens when we start getting hypoxic, the mitochondria start to swell, the membrane starts to bleb or break off, and you get loss of the microvilli structure at the surface. So that's hypoxia. Here's another example of a type of cell injury. This is cell injury that's caused by free radicals. Free radicals are chemicals, compounds that have a single unpaired electron in an outer orbit. Now the problem with free radicals is they're very reactive. They can initiate autocatalytic reactions that convert other molecules into free radicals. So if you have one free radical, it can kind of multiply and convert other free radicals. And these free radicals can break down and damage cellular structures. Now, free radicals are generated by a host of normal processes. For example, normal metabolism can create reactive oxygen species, oxygen-containing free radicals. Immune cells, such as macrophages, actually use free radicals to break down things in their lysosomes. So inflammation can cause the generation of free radicals. Chemical injury, for example, things like carbon tetrachloride, generate free radicals. Ionizing or UV radiation causes free radical formation, and certain drugs can cause it as well. And the last thing is reperfusion injury. If you have a hypoxic injury and all of a sudden you get reperfused, that reperfusion causes an influx of oxygen, which leads to the formation of oxygen-reactive species or free radicals. The most common free radicals are things like superoxide anion, hydrogen peroxide, hydroxyl ions, and nitric oxide derivatives. Now, free radicals do three major things in a cell that are bad. One is that they peroxidate membranes, and this causes a weakness of cell membranes, can cause leakage of cell membranes, general dysfunction of cell membranes. The other thing that happens is you get oxidative modification of proteins, which causes the proteins to stop functioning properly. The last thing you can get is genetic damage. You can get reactions with thymine, leading to thymine dimers or single-stranded breaks in DNA, which lead to genetic damage to the cells. Now, this propagation, this free radical generating additional free radicals, often drives these reactions. Now, how do we get rid of free radicals? Well, we have some antioxidants in our cells. We have some natural antioxidants, things like vitamin A, vitamin E, and vitamin C, which can convert free radicals back to their normal form. We also have a bunch of enzymes which protect us from free radicals. And these include catalase, superoxide dismutase, and glutathione peroxidase. Catalase is responsible for decomposing hydrogen peroxide. Superoxide dismutases convert superoxide to hydrogen peroxide, which can then be broken down by catalase. And glutathione peroxidase catalyzes the conversion of hydrogen peroxide to water using a sulfhydryl group from glutathione. You can also get free radicals can spontaneously decay as well, though that obviously isn't the most efficient way to get rid of them. So here's an example of generation of free radicals, and you can see on the right that inflammation, radiation, oxygen toxicity, chemicals, reperfusion injury, etc. generate these free radicals. If they are not dealt with, then what you get is what you see on the bottom left. These reactive oxygen species peroxidate membranes, cause DNA fragmentation, cross-link and fragment proteins, and you get cellular dysfunction. Now, on the other hand, if you have vitamin E, A, beta-carotene, superoxide dismutase, glutathione peroxidase, vitamin C, glutathione, so on and so forth, you're able to deal with those, break down those reactive oxygen species, and protect the cell from damage. Now here's a schematic of how these free radicals are broken down. Here's the superoxide free radical right here. broken down by superoxide dismutase to form hydrogen peroxide. Hydrogen peroxide then can be broken down by catalase into water. Okay, but hydrogen peroxide can also be broken down into hydroxyl radicals, and that occurs in a thing called the Fenton reaction, where you have free iron in the cells. In the ferrous state, which is 2+, is converted to ferric iron, 3+. It's oxidized, and in the process, the hydrogen peroxide is reduced to hydroxyl radical, which can damage cell membranes and so forth. And so to deal with this hydroxyl radical, we use glutathione peroxidase, which takes glutathione molecules and using the reducing power of their sulfhydryl bonds, converts that hydroxyl radical back to hydrogen peroxide such that it can be converted by catalase to water. And so this is the whole process by which we generate and break down free radicals. Another example of cellular injury is chemical injury. Now there are two mechanisms of chemical injury. One is direct. You can get a direct chemical injury to cellular structures. For example, mercury injury. Mercury binds the sulfhydryl groups of the cell membrane, increases permeability, especially in the cells of the GI tract and kidney. So it causes direct damage to those cells. Another example is cyanide. Cyanide affects the cytochrome oxidase, part of the electron transport chain. It blocks oxidative phosphorylation, causes direct damage to the cell. There's also an indirect mechanism where certain chemicals are metabolized, certain chemicals that are by themselves inert are catalyzed to toxic metabolites. For example, carbon tetrachloride on its own is not that harmful, but it gets converted by a P450 enzyme, a cytochrome P450 enzyme, to carbon trichloride, which is a free radical, which then can damage cells. And so some chemicals that are by themselves inert are metabolized to things which can be damaged, and that's an example of indirect chemical injury. So here's an example. This schematic simply shows that process of carbon tetrachloride breakdown to carbon trichloride radical and how that affects different parts of the cell.